The lecture titles is Nucleic Acid, Part 2, Structure and Function. The chromatin of a human being contains from 46 chromosomes, DNA molecules associated with protein. This DNA strand associated with a protein, this protein called histone. DNA called around core particle, histone protein. Zigzag loops and called on itself. DNA copies itself to form two parallel sister chromatin. This is chromosome and this is the strand of DNA called with particle called histone. Histone is a protein found in nucleus. Why DNA is double strand? The X-ray diffraction data analysis that show DNA as a double strand with observation of Jarkiv that found DNA molecules, the concentration of deoxyadenosine A nucleotide equal that of thymidine nucleotide, A equal to thymidine. And the concentration of deoxyguanosine G nucleotide equal that of deoxycytidine C nucleotide. So concentration of guanidine equal to cytidine. That suppose that Watson, Crick, and Wilkins proposed that early 1950 model of double strand to the DNA. Example on this, if the concentration of adenosine A equal to 14% in DNA sample isolated from virus, what are the concentration of guanosine, cytidine, and thymidine in this DNA sample? So concentration of adenine equal to thymine, so thymine equal 14%. So concentration of guanine plus cytidine equal 100% minus adenine plus thymidine. So 100 minus 14 plus 14, 100 minus 28 equal 72. 72 because it's guanine equal to cytosine, so 2 guanine equal 72. 72 divided by 2 equal 36% concentration of guanine and concentration of cytidine. This is Watson and Crick. This is X-ray diffraction data of DNA. Found the structure, double strand structure of DNA. This is also Franklin and Wilkins work on the DNA structure. The double strand is stable because it contains hydrogen bonding. The hydrogen bonding in the DNA is between thymidine and adenosine. There is 2 hydrogen bonding between thymine and adenosine, and 3 hydrogen bonding between cytosine and guanosine. This is hydrogen bonding. So the double strand stable with hydrogen bonding between nitrogen base. This is adenine thymine, thymine adenine, cytosine guanosine contain hydrogen bonding. The double strand DNA contain major groove and minor groove. The distance between major groove is 34 angstrom. In the next slide, the DNA double strand unwinding of DNA by helical expose the DNA base replication fork. So the replication takes place with hydrolyze ATP, hydrolyze the double strand, and the order break hydrogen bonding between the double strand. and a new strain of DNA are formed. So the nitrogen base, it is represent the genetic code. If we have the mutation on the single amino acid substitution in a protein, cause, as example, sickle cell disease. We have the normal DNA sequence of nitrogen-based DNA. We have cytosine, thymidine, cytosine, guanine, adenine, guanine. This is the normal code, will translate to the glutamic acid in the protein of hemoglobin, In the mutation, we change the thymine with adenine, so the anticode will be guanine, thymidine, guanine. This is code for the valine amino acid. Valine amino acid will change this normal glutamic, the sequence number 6 amino acid, glutamic will change to the valine. This mutation, valine, will give me sickle red blood cell. This is example on the mutation in the nitrogen base. So we have DNA and RNA. There are a difference between RNA and DNA. The RNA, ribonucleic acid, contain nucleotide, contain ribonucleic acid, contain ribonucleotide linked together by 3,5 phosphodiesterase bridge, analog to this in DNA. So DNA and RNA have several specific differences. Number one, in DNA, in RNA, the sugar moiety is ribose rather than 2-deoxyribose of DNA. Number two, the pyrimidine compound of RNA differ from these of DNA. Also, RNA contains the ribonucleotide of adenine, guanine, and cytosine. It does not possess thymidine instead of thymine. RNA contains the ribonucleotide of uracil. So in both DNA and RNA, we have 4 nucleotides. In RNA, we have adenine, guanine, cytosine, and uracil. In DNA, we have adenine, guanine, cytosine, and thymine. Number 3, RNA exists as a single strand, whereas DNA exists as a double strand helical molecules. The single strand of RNA is capable of fold back on itself like a hairpin and thus acquiring double strand characteristic. Number 4, since the RNA molecules is a single strand, it is guanine content does not necessarily equal to cytosine content, nor does the adenine content necessarily equal to uracil content. Number 5, RNA can be hydrolyzed by alkali to 2,3 cyclic diester of the mononucleotide compound that cannot be formed from alkali-treated DNA because of the absence of 2-deoxy group or the alkali. The ability of the RNA is useful both diagnostically and analytically. We can distinguish between DNA and RNA by alkali solution. The RNA dissolve and form 2,3 cyclic diester, and DNA cannot dissolve in alkali solution. So the structure of RNA is single strand, and the DNA double strand. Both of them formed from nucleotide. Nucleotide in RNA, ribonucleic, contain ribose, and DNA, deoxyribose. There are many types of RNA. Number A, messenger RNA, mRNA. This class is the most heterogeneous in size and stability because it is synthesized from DNA in the nucleus and depends on the type of protein we need to translate. So it is so different in size and stability. Their function as a messengers, conveying the information in gene to the protein synthesis machinery. Each serves as a template on a specific sequence of amino acids to polymerize form specific protein molecules. The ultimate gene product, synthesized from nucleus, transport to cytoplasm to the ribosome to send the message to translate the gene to the amino acid. So this is DNA. We have the mRNA. This is called transcription for an mRNA. mRNA leave the nucleus to the cytoplasm to the ribosome, and this is ribosome, and this is protein synthesized from mRNA to form full protein molecules. Number B, transfer RNA, tRNA. tRNA molecules vary in length from 74 to 95 nucleotides. So it is contained from 74 to 95 nucleotides. The tRNA molecules serve as adapter for the transfer. So the tRNA transfer the amino acid. So transfer amino acid to the mRNA to inform the sequence of nucleotide of mRNA to specific amino acid. So there are at least 20 species of tRNA molecules in every cell, corresponding to each of the 20 amino acids required for protein synthesis. Each amino acid had one tRNA to transfer amino acid. All tRNA molecules allow extensive folding and interstrand complementary to generate secondary structure that appear like a clover leaf. This is the structure of tRNA. We have 4 arms for tRNA. This is 3 dimension of tRNA. We have, this is amino acid. This is tRNA for amino acid leucine. So leucine bond here, and this is arm for anticodon on the mRNA. This is codon and anticodon of mRNA translate the gene to the amino acid leucine, and the second codon to the another amino acid to form peptide bond, and so on. We have ribosomal RNA, rRNA. The type C, ribosomal RNA, rRNA. A ribosome is a cytoplasmic nucleoprotein contained from nucleotide and protein, nucleoprotein structure, that act as a machinery for the synthesis of protein from the mRNA templates. On the ribosome, the mRNA and tRNA molecules interact together to translate into specific protein molecule information transcript from DNA, the gene source. The component of mammalian ribosome, which has a molecular weight of about 4.2 10^6 and sedimentation velocity of 80 s Svedberg unit. So, the mammalian ribosome contains two major parts, nucleoprotein, we call subunit, large subunit and small subunit. Large subunit, the molecular weight is 2.8 10^6, 60 s, and small subunit molecular weight is 1.4 10^6, 40 s. So we have two subunits, large one 60 s and small unit 40 s. Number D, small stable RNA, ssRNA. A large number of described highly conserved and small stable RNA species are found in eukaryotic cell. The majority function of these molecules are complexed with protein to form ribonucleoprotein, also contain ribonucleotide and protein. They are distributed in the nucleus, in the cytoplasm, or in both. In the nucleus for the synthesis of mRNA, and in the cytoplasm for the synthesis of ribosome. Their size range from 90 to 300 nucleotides. Also, there is a small nuclear RNA, snrRNA. Subset of this RNA are significantly involved in mRNA processing, also contribute in the mRNA processing and gene regulation. The reference of this lecture is Harper Illustrated Biochemistry or any book biochemistry contain nucleic acid chapter. Thank you very much. Is there any questions?